Hi guys, Jason Matthew here again, and today we're going to be looking at lipids, and we're doing part 2. So we're going to be looking at membrane lipids. So please, if you haven't looked at part 1 as yet, go to the BiochemJM channel and look at part 1, because part 1 laid the foundation for this part. So you're going to better understand this part if you look at part 1 first. So just to recap, in last class, which would have been lipids part 1, we looked at structures of some common lipids, functions of lipids. We looked at saturated versus unsaturated fats. We named fatty acids. We learned how to use the delta designation, as well as how to label it via the omega nomenclature. We looked at trends in melting point in fatty acids. We were able to look at the 2 essential fatty acids, omega fatty acids in general, what are trans fat. We looked at the chemistry behind tag formation, and we also looked at the hydrolysis of fats. So if you are going through this list and you're seeing something here that you're not familiar with, well, then you definitely need to go back to the BiochemJM YouTube channel and look for lipids part 1 and go through that before you continue with part 2. Now, another thing that I've said before, but I just want to say it again, is that these videos are not the end or the only source that you should be using for your studies. I will always urge you all to go take up your textbooks, go in the library, go on the internet and so on, and get as much information on the topics as possible. Because only when you look at a topic from different angles will you truly appreciate what you're learning, and it's going to make it easier for you to learn, and you're going to see it in your grades and so on. Alright, as well as when you're looking at my videos, I want you to have some paper and just take notes, because the real way to learn is that you need to write. You need to draw these diagrams. You need to learn to write, because when you're in the exam room, it's just you, a pen and a paper. So get in the practice of writing and drawing these diagrams. It will be beneficial to you in the long run. So, and remember, if you think this video is too long or it's too fast paced, always remember there's something called the pause button. So when you think you have had enough, just press pause, take a break, come back at it, and you can also rewind. So learning is totally in your hands right now. Alright? So let's go and see what we're doing for part 2. So for today's class, we're going to be looking at the structure of membrane lipids. And in particular, we're going to be looking at phosphatidates, sphingosine, and cholesterol. And finally, we're going to be looking at how cholesterol affects fluidity of membranes. So these are some typical membrane lipids, and we have looked at triacylglycerols in part 1. So today we're going to be looking at the glycerophospholipids, sphingolipids, and if we have time, we might look at galactolipids. We'll see. So this is our familiar friend here, glycerol. We looked at this in part 1. And again, if you need to describe the glycerol molecule, it has 3 carbons, and it also has 3 hydroxyl groups, which are these OH groups. And if you remember from part 1, at each hydroxyl group, a carboxylic, or I should say a fatty acid via its carboxylic group is able to react, right, via condensation reactions. So you're going to be losing water, and an ester linkage will be formed. Again, this is what we did in part 1, so please go back and look at it if it's sounding a little unfamiliar to you. So all we have done in this diagram is basically added a phosphate group to the glycerol molecule. So again, this here is your glycerol molecule, and they have numbered the carbons 1, 2, and 3, and this phosphate group has combined with the OH group that was on carbons 3 of the glycerol molecule. So that's why we call it glycerol 3-phosphate, because the phosphate group is being added to carbons 3 of the glycerol molecule. So here is a typical or general structure of a glycerophospholipid. So just to point out, all glycerophospholipids will have glycerol as its backbone. Alright, so that's why we call it a glycerophospholipid. Now, this here is your glycerol molecule. So you can see that. Alright, and what has happened is that so that what I have just boxed off there, that's glycerol. That's the glycerol backbone. I remember on glycerol there's 3 OH groups. So on carbons 1, the hydroxyl group on carbons 1, a fatty acid combines with that one. Then a fatty acid combines on carbons 2, and a phosphate group combines on carbons 3. Alright? Now, these two linkages here on carbons 1 and carbons 2 is what, when a carboxylic group combines with the hydroxyl group, you're going to get an ester linkage. So you have 2 ester linkages here, one on carbons 1, one on carbons 2. And this here, what do you think you call this guy? You probably saw it when you looked at DNA. When the phosphate combines with the OH. That's right, it's a phosphodiester group. Alright? Now, what is the difference between these two fatty acids? If you look closely, you should see that this fatty acid, this hydrocarbon tail, has a double bond. So it's unsaturated, while in this one, it's saturated. It doesn't have any double bonds. And that's a general thing, in that in most, if not all, glycerophospholipids, a saturated fatty acid is going to combine with the OH group of carbons 1 of glycerol, and an unsaturated fatty acid is going to combine with the OH group on carbons 2 of glycerol. And on carbons 3, you'll have the phosphate group. And if you watch here, there's an X. And basically what that X means is that other groups could add on here. Now, if X is H, which is hydrogen, if X was H, we will call this molecule a phosphatidate. But right now, what we're looking at is the general structure of a glycerophospholipid. So remember, as I said, let's just go back a little bit. You see this X? This X will represent several groups. So as I said before, if X is hydrogen, it will be phosphatidic acid, or we call phosphatidate. Now, if instead of hydrogen, the X group was a choline group, then it will be phosphatidylcholine, and so on. So you can look at, these are some of the common glycerophospholipids present. So phosphatidate, just to recap, you need to be able to draw as well as describe the structure. And all phosphatidates have a glycerol backbone. There's a saturated fatty acid at C1, an unsaturated fatty acid at C2, and a phosphate group at C3. Now, a phospholipid, or glycerophospholipid, is amphipathic, meaning that it has both a hydrophilic group as well as a hydrophobic group. Hydrophilic meaning that it likes water, hydrophobic meaning it doesn't like water. Hydrophilic or polar, hydrophobic, non-polar. So in a glycerophospholipid or in a phospholipid, there's normally a polar head. Alright, so this here will be a polar head. And then you have your two tails, the two hydrocarbon tails, which are hydrophobic. Alright? Now the only thing that we could add here additionally is that if you look here, you see there's a bend there. Can you reason out why there's a bend? Why would this hydrocarbon tail change direction? So if you watch it here, there's a bend. What causes that bend there? So I hope you rightfully deduce that this is carbons 2. And on carbon 2, there's an unsaturated fatty acid. And when we say unsaturated, we mean that there's a double bond present. And to be more specific, a cis double bond. And what a cis double bond would do, it would change the direction, it would form a kink, and will change the direction of the hydrocarbon tail. So if you see it bends somewhat. This tail here is straight, and the reason for that is because this is a saturated hydrocarbon. So there's no kinks present, but on carbons 2 there's a unsaturated fatty acid joined to it. So the there's a kink and it changes direction. And just to recap and to reinforce, because it's a naturally occurring fatty acid joining this, the double bond is a cis double bond, and a cis double bond causes a kink in the hydrocarbon tail. So just be very careful. And these two hydrocarbon tails will be hydrophobic, while up here the head will be hydrophilic. It will like water. And when we look at membrane structure next, and when we look at the lipid bilayer, you'll see that how important that fact is. So let's remember, virtually all naturally occurring unsaturated fatty acids, their double bonds will be in the cis configuration. But we saw in the first lecture that how we could also have the trans configuration. Now the trans configuration, it doesn't form any kinks. It doesn't change the direction of the fatty acid. So if you see this one is straight, while in the cis fatty acid, you see that there's a kink. The fatty acid tail bends. Alright? Unsaturated fatty acid will have a similar shape to a trans fatty acid in that there's no kinks present, there's no bending. But for cis fatty acid, meaning a fatty acid with a cis double bond, there is bending. There's a kink being formed. Right? So kinks, they cause these fatty acids to pack less. There's no tight packing, and because of that, they are more fluid and they're good for membranes. Right? Trans fatty acids are bad. Right? There are no kinks. They pack like tightly, and there's an increase in LDL, which is bad cholesterol. All right, and this has been related to heart disease, you know. So be very careful of trans fat. In fact, you should try to avoid trans fat in your diet like the plague. It's very bad for you. Right, so now we go into sphingolipids, which is a different type of lipids found in the membrane. Now, with sphingolipids, they do not have a glycerol backbone like their glycerophospholipids brothers. They have what is called a sphingosine backbone. Alright? So this is a structure here of sphingosine. So what I want you all to do now is that same pen and paper that you're supposed to have with you, and you're supposed to be writing copious notes right now. I will tell you to describe. So I have drawn sphingosine. What you should do is you should draw sphingosine, because I would, at least my students, I would require that you are able to draw these things. I want you all to describe what you're seeing right now. And you know what? I'm going to be a good sport. I'm going to number these carbons for you, so you could actually see what they are. So this is carbons 1, carbon 2, 3, 4, 5, and well, I guess you get the picture. Continue. And tell me describe this molecule. Alright, tell me what functional groups are present, how many carbon atoms there are, where is the double, I'm seeing a double bond, that kind of thing. So try to look at a molecule and describe it as fully as possible. Alright, so you should have a pen and paper and you should start watching this molecule. First, draw it, alright, and keep practicing drawing for the exam, as well as also look at it and try to be able to describe it for me. So jot down some descriptions for me, and then we'll go to the next slide. Alright, so sphingosine, let's see. You should have said that it's on branch. And if you didn't say on branch, that's fine. But you should at least say that it had 18 carbons. And it's an alcohol because there's an OH group present. So you should at least tell me, you should have had on your paper that when you counted up the number of carbons, you got 18 carbons. You might not know that it's a trans double bond, but the double bond is of the trans configuration, and it's between carbons 4 and carbons 5. There was an amino group at C2, carbon 2. And there are two hydroxyl groups, one at carbons 1 and one at carbon 3. Now, if sphingosine combines with a fatty acid. So the fatty acid is in yellow here. Sphingosine is in this light pinkish color. Alright, now if there is X here, then we say it's a sphingolipid, where X could be many things. But let's say if X was hydrogen, then this whole molecule would be called ceramide. Now, what I want to ask you, some chemistry now, and something that we came across already, but I just want you all to tell me what you would name this bond. Alright, is it a ester? Is it a glycosidic bond? What is it? Tell me what is the name of that. So try to describe this sphingolipid to me. Because basically, if you already described sphingosine, you're 90% there. It just now you need to add in this fatty acid. This fatty acid is combining with what carbon on sphingosine and what type of bond is being formed. The other thing that you should always remember is that for a sphingolipid, the backbone is sphingosine. So you need to be able to describe sphingosine, which we have done before. Alright, so as I said, let's describe ceramide. And again, it's a sphingosine backbone, and we have already described that. And the only thing new here is that the fatty acid group, right, of the fatty acid is linked to the carbon 2 amino group of sphingosine by an amide bond. So let's go back. This here that we had already boxed off before, this is an amide bond. So the fatty acid would have combined with the amino group on carbon 2 of sphingosine to form an amide bond. So that's what we have here. Right, now remember when we looked at the general structure of sphingolipid, we had an X group. Now if X was hydrogen, it would be called ceramide. And you can look at the others, but I just want to point out sphingomyelin, because that's when you have, when X is phosphocholine, you have sphingomyelin. And you should at least have heard the word myelin before, I hope. But myelin, or sphingomyelin, is a key lipid found in the membrane of the myelin sheaths that insulate the axons of nerve cells or the neuron. All right, and you could do some reading about it because it's very interesting stuff. And that is these myelin sheaths, they increase the speed at which nervous impulses conduct themselves. And it's extremely important because there are some diseases that break down sphingomyelin and a lot of bad stuff happens. So you can do some reading. Read up about multiple sclerosis as well, and you'll see some serious things happening there. Now, if the X group is a glucose molecule, then we say it's a cerebroside, glucose cerebroside. And if it's an oligosaccharide attached as the X group, then we say it's a ganglioside. So let's be familiar with the term ganglioside, cerebroside, sphingomyelin. Alright, so as I said before, sphingomyelin is the key lipid found in the myelin sheath. Alright, so that's just some more structures of it. Now the last lipid that is found in the membrane that we will be discussing in this lecture will be cholesterol. And this molecule, I would expect my students at least to be able to draw cholesterol, and also not only just draw it, because anybody could draw the cholesterol, but I also want you to describe it to me. Right? And we will see some better diagrams along the way. So let's use this one. This is a nice one. This was taken from the Leninger, I believe. Now you see there's this small hydroxyl group on carbon 3, and that's the polar head. Alright, you have 4 rings, and then you have a flexible hydrocarbon tail. So what I want you all to do is draw this on your piece of paper, on your notes, and then you know the drill. I want you to describe this cholesterol molecule as best as you can. So tell me where the double bonds are, where the hydrocarbon is, where the hydroxyl group is, how many carbons, that kind of thing, and see what you come up with. Right, so let's go do it together now. So for cholesterol, there are some main points I want you to always remember about the cholesterol molecule. And what you could do is that if you had followed my instruction, you would have drawn the cholesterol molecule, combine it, and make sure you identify from your structure what I'm saying here. First thing is the steroid nucleus. Very, very important. It's made up of 4 fused rings. And all 4 rings are not identical. Look at the shapes and stuff. Count the number of sides. You will see that 3 rings have 6 carbons. And we label the rings A, B, and C. And then the 4th one has 5 carbons, and we label that guy D. There's a hydroxyl group at C3, and that's the polar part of cholesterol molecule. And then there's an alkyl side chain, a flexible hydrocarbon tail at C17. Alright? And there's some other stuff that you could describe about cholesterol, like where the double bond is and so on. So I'll leave that up to you. And I didn't state here how many carbon atoms, but by now you should know the drill. Right. Now, do you need to learn any of these structures? No. But I would hope that at least that some, if not all of these names should sound or should be familiar. Testosterone, especially, right? You should know what testosterone is. You should have heard aldosterone. Cortisol, maybe prednisone. If you are an asthmatic patient and you have been in an emergency ward where you've been nebulized, you may have heard of, you may have been given prednisone or prednisolone. Alright, so all these here, they have one thing in common, and that is this steroid nucleus. 1, 2, 3, 4 fused rings. And the reason why this slide here is up is not for you to know these names or to even draw these things, but the one thing I want you to appreciate is that they got their steroid nucleus from cholesterol. So in other words, the body would take the cholesterol molecule and make these guys. And all of these guys here, testosterone and so on, they fall under the class steroid hormones. So all steroid hormones will have these 4 fused rings, which we call the steroid nucleus. And they got this from cholesterol. So cholesterol is a precursor molecule for the formation of steroid hormones such as testosterone and so on. Right, so back to the cholesterol molecule. Cholesterol is found in the membranes and is particularly abundant in animal cell membranes. And for it to be present in the membrane, the cell membrane, it has to interact with phospholipids. Now, phospholipids, as we saw earlier, had a hydrophilic head and a hydrocarbon tail. And what the cholesterol would do, it will orient itself in the membrane that its hydrophilic head will interact with the hydrophilic head of the phospholipid, and its hydrophobic portion, its non-polar portion of cholesterol, is going to interact with the non-polar portion of the phospholipid. So it's very happy and comfortable where it is in the membrane. So each cholesterol molecule orients itself in the lipid layer so that its single hydroxyl group is close to the polar head group of a neighboring phospholipid molecule, where it forms a hydrogen bond with the oxygen of the ester bond between the glycerol backbone and a fatty acid. Right? And that is basically what I just showed you in the diagram, right? Remember, for cholesterol, it's the hydroxyl group is the polar head, and it's going to interact with the polar head of the phospholipid group. Simple stuff. The steroid rings and hydrocarbon side group of the cholesterol molecule interact with adjacent hydrocarbon chains of the membrane phospholipids. So the bottom line is this. The hydrophilic portion of the phospholipid is going to interact with the hydrophilic head of cholesterol, which is the hydroxyl group, and the hydrophobic portion, the non-polar portions of the phospholipid is going to interact with the non-polar portion of cholesterol. Now, cholesterol plays a big role in maintaining fluidity of cell membranes. And as I said, cholesterol is widely abundant in animal cells. Now, what you could do some homework and you could write back and tell me, is it I mean, I know definitely it's not frequent, but is there any cholesterol in plant cell membranes? I am not sure. I'm tempted to say no, but in biology, biochemistry, there's no such thing as definitive no. So, guys, please do some extra reading. I don't know if you'll have it in your notes or what, but you can give me some feedback and let me know if there are any plants that have cholesterol in their membranes, and that'll be interesting to find out. But I know it's definitely abundant in animal cell membranes. So cholesterol is wedged between phospholipid molecules in the plasma membrane of animal cells. At warm temperatures, so it's two things could happen. Either the temperature is warm or it could be cold. And in both of them, like in a warm temperature, it means that there's a lot of heat energy around. So there's a lot of kinetic energy. And what could happen is that the phospholipids in the cell membrane, they will want to move a lot. They have a lot of kinetic energy, so they will want to move. But if they move a lot, it will destabilize the membrane. So what happens there is that cholesterol will restrict such movement because of how it's wedged between the phospholipids. It's going to restrict that movement and therefore reduce fluidity. Alright? So it's reducing fluidity when it's warm, because at warm temperature, because of that, all that kinetic energy from that heat energy, the phospholipids, they want to move. So they want to reduce that. Now the opposite will happen if the temperatures start to cool down, because at cool temperatures, there's less kinetic energy, so the phospholipids, they start to move less. And what can happen there is that if there's no cholesterol present, they will pack tightly because they're moving less. They're going to start interacting with each other. They're going to tightly pack, which will make the membrane lose its fluidity now, or some of it, and that could be bad as well. So at cool temperatures, cholesterol maintains fluidity by preventing this tight packing of the phospholipids. So again, it does it differently according to how the temperatures. At warm temperatures, it will reduce fluidity by restricting movement of the phospholipids, and at cool temperatures, cholesterol is going to maintain fluidity by preventing tight packing of the phospholipids. Alright, so we have come to the end, basically, of lipids part 2. But I've been looking at the YouTube channel and we have been getting a lot of international visits. So I'm very grateful for that. So we're going to give you a little treat. Just give you a little taste of the kind of enormous talent that this country have. So for Carnival 2012, Carnival Monday, Carnival Tuesday just passed. It was 20th of February, 21st of February. And Marshall Montano, he's our soca monarch, groovy soca monarch. And he also won Road March. And he's a very talented guy and he has been all over the world. So I don't know, maybe you all have been even to some of his concerts. But for our international people, I'm going to give you a little taste of the kind of talent that we have. And please, just take it in. I was going to give you a little short little clip. Right, so as you can see, right, his song is Mr. Fett by Marshall Montano and is directed by Peter C. Lewis. I have no connection with this video. I have not done anything to this video. It's on YouTube, so you can always check it out there. But I'm just proud of our local guys and I'm just showcasing it to our international peeps out there. So please have a little listen and you can always get the rest of the song on YouTube. Right, so as I said, just a short taste. I mean, go to YouTube, you're going to get the whole video there, and enjoy yourself. I'll put it as a link on my channel as well, so you can always see it from there as well. You know, so excellent stuff going on in Trinidad. So as usual, if you haven't joined our Facebook pages yet, please do a search for BiochemJM. We love to hear from you all. If you need to email me, that's my email address there. And as usual, guys, thank you very much for listening. I hope I help you all out. And best of luck.